So welcome back to the Classics of Immunology Journal Club. And we've been moving along now for quite some time, since 1798 and Edward Jenner. Now we're up to 1985, and we're going to review a paper by Emile Unanue, who we reviewed another paper, a prior paper by him that I'll get into in a second. The other authors on this paper were Bruce Babbitt, Paul Allen, Gary Matsueda, Edgar Haber, and then Emile Unanue. And the title of the paper is Binding of Immunogenic Peptides to IA Histocompatibility Molecules, published in Nature in September of 1985. Before we get started, I just want to remind you that if you like the video, then go on the YouTube side and like it, and then click on the subscribe to the videos, and then click on the bell, and you'll get notified when I make another one, usually about once a week. And also go into my website, and you can find all the classical papers, and you can read them for yourself and see whether or not I'm telling you the straight scoop or whether I'm not telling you the straight scoop. Emile Unanue has a very interesting history, and I went into it a little bit before. This is sort of the background of this paper. In 1985, he'd just been recruited to come to Washington University in St. Louis from Harvard, where he was a younger faculty member of Baruch Ben Esraf's laboratory. And Baruch was the head of pathology at the Dana-Farber Cancer Institute at that time, and also professor of pathology and head of the Harvard department. So Emile was from Cuba, and he graduated from Havana Medical School in 1960. And of course, if you know your history, January 1st of 1959 is when Castro rode into Havana and liberated the city and took over, essentially. So it must have been heady times in Cuba at that stage of the game. Emile, he got out right after he graduated, and he got a job with Frank Dixon, who was one of the glitterati in immunology at the time, who was at the University of Pittsburgh. And you might remember that the University of Pittsburgh is where Jonas Salk did his pioneering studies with the polio vaccine. Anyway, soon after Emile got there in Pittsburgh, Frank Dixon moved to the Scripps Research Clinic and Research Foundation in La Jolla, California. Much better climate out there. Emile's, and much more like Havana, probably. So Emile must have been very happy. And he spent a couple of years working as a postdoc with Frank Dixon, and then he got a second postdoc with Bridget Askenis, Ida Askenis, at Mill Hill in London, and that was where he did the work that I reviewed a while ago. Actually, it was in May of 21, a paper that came out in 1968. So that was 17 years ago. So now you see the progress of this particular person from postdoc to chair of the pathology department at Washington University in St. Louis. And with Askenis is where he started his work on what became called eventually as antigen processing and presentation. And the paper that he did that I reviewed before was showed that macrophages seem to hold, if you fed them antigens, macrophage would hold on to the antigens for quite some time. I think it was a couple of weeks where he was able to demonstrate these macrophages were still antigenic, immunogenic if he transferred them and so forth. The intro of this paper, it was known, and this is what they go into in terms to set the background of their paper. At that time in 1985, it was known that in order to activate T cells so that they would proliferate and make other cytokines and so forth and participate in an immune response, you needed to have antigen presenting cells. And they came to be called professional antigen presenting cells if they could present antigens to the T helper subset of cells, the so-called the CD4 positive T cells. And of course, Alice Reinhertz had shown back in 1982 and 83, and we reviewed those papers, that helper T cells had on their surface a molecule that labeled them as helper T cells, and as CD4 was the name of the molecule. It also, at the time that Ellis had shown, and we reviewed that paper, is that the so-called helper T cells could also be killer cells, and so don't forget that. That's very, very important. Anyway, so it was known that you needed these antigen-presenting cells. It was known that on the surface of these the professional antigen-presenting cells that activated the T helper cells, that they had to be histocompatible. The macrophages had to be histocompatible with the T cells. That is, they had to have the gene products of the major histocompatibility locus, MHC complex, that would code for the molecules that then was placed on the surface of these macrophages, and also B cells. They were the two professionals antigen presenting cells. And it was known that they had to be there. And it was thought that primarily because of Ellis's work, as well as others, that somehow the T cell would recognize that MHC gene product. And it was also thought and known at the time that the other antigens, the so-called the peptide antigens, were processed by these T cells. But it wasn't known how these MHC encoded molecules did whatever they did. And so by the time that Emil Unanue was the entire time he was at Harvard, and then when he migrated to WashU, other people had tried to just incubate antigen presenting cells with antigen to see if the antigen somehow stuck to the antigen presenting cells. Seemed like a logical thing to do, but they couldn't show that phenomenon happened, and they couldn't show specificity and so forth. And so it was sort of a conundrum at that stage. And so what this paper set out to do was to determine whether or not you could use purified MHC encoded molecules, get rid of the rest of the cell, take the membrane of the cells, of the presenting cells, purify away everything else, and have purified MHC molecules, encoded molecules, and to see if they could bind the peptide. And so that's what they wanted to find out, and that's what they did. And the thing is that this, like all the Nature papers, this is one of these classic Nature papers that's only 2 pages. It's sort of like the Watson and Crick's 1953 paper of the structure of DNA. It was only 2 pages. And so the ambition of all young people back in the day was that if you could get a 1 or a 2 pager into Nature, it doesn't get any better than that. And the materials and methods. And the other thing about this is, even though it's only two pages, there's a lot of work, a lot of background work that went in to be able to do the experiments that they present in this paper. And so what they did was they made B cell hybridomas from the right histocompatibility strain of mouse. And the reason they wanted hybridomas was so they could grow up huge numbers of them. And they grew up 10 to the 11. So that's 100 billion. They then broke them open with nitrogen cavitation, and they purified the membranes, and then they purified then they solubilized the membranes, and with an monoclonal antibody to the MHC encoded molecule, they made affinity columns so that they could take these solubilized membranes and pass them through the affinity column. Everything else and then wash the column, and everything else would go through, and then they could elute the MHC encoded molecule from the antibody that was bound to Cephalus beads, usually by changing the pH, so that then you could get purified molecules. And they showed then by STS Page that they were pure. There were only 2 bands, was what they expected, because that work had already been done. And they also had done work on trying to isolate specific peptides. One of the things I forgot to tell you is that the other thing that was sort of strange about T cells, that unlike antibody molecules that would recognize the three-dimensional structure of a molecule as the antigen molecule, T cells recognized strings of peptides of only anywhere from 6 amino acids to 10 amino acids. And so T cells would recognize distinct sequences and chunks of the molecule. So that was what happened in the antigen processing part. And the antigen presentation part is what this paper was focused on. And they'd already done a lot of work with an antigen, a protein antigen called hen egg white lysozyme, or HEL, and it had been used by many people in immunology as a really good antigen for mice coming out of a chicken. And so they had a peptide that was like 10 amino acids long, and it was amino acids number 46 to 61 in the sequence of HEL, and it was specific it was antigenic as long as you fed APCs this particular peptide. The other thing they did was that they developed T-cell hybridomas that would recognize this peptide, and they used the T-cell hybridomas as their readout cell to see if the T-cell hybridomas would get activated. And then they derivatized the peptide antigen with a fluorescent molecule that was abbreviated NBD, and the reason they did that was that gave them the detection device to detect any antigen of the peptide, whether it was bound to the MHC encoded molecules or not, and so forth and so on. And then they did equilibrium dialysis, and that's when you put a large protein inside of a dialysis tube and then add the antigen as a small molecule into the mix, and then they would incubate that mixture of the two molecules, the receptor molecule and the ligand, together for 72 hours at 37 degrees, and then you put that into a dialysate with a larger volume, and you just put it in magnetic stirrer and you stir it around, and the smaller molecule can equilibrate on both sides of the dialysis membrane, and on the inside of the dialysis tube is the receptor molecule, which in this case was MHC encoded molecules. And that was a classic way that the immunochemists had done their experiments over the years to determine the affinity of an antibody for an antigen and so forth. That was all pretty straightforward, but of course that was a technique that you had to learn. And so I think that Emil Unanue benefited a great deal from being at Harvard, where he could learn all these sophisticated molecular techniques. That's what they did. And so that the results, I'll show you by screen sharing. So this is the paper that came out in September of 1985, and this is figure 1. And as it says here in the caption, this is the response of the T-cell hybridoma cells to either native peptide or peptide bound to the fluorescent molecule NBD. And so here, and this is what I like about this paper, that they used interleukin-2 as their readout as to whether or not the T cells were activated or not. And so on the y-axis, they've got tritiated thymidine incorporations into the assay cells, which are the CTLL. They would incubate the T cell hybridomas with the antigen, different concentrations, very, very low up to very, very high concentrations. And this is the NBD molecule here that fluoresces when UV light hits it. And this is the peptide right here. And this is in the solid circles is just the peptide itself. And this is the one that's derivatized with the NBD. And you can see that, if anything, the NBD derivative is even more potent and activating the T cells to make interleukin-2. And of course, then what they did was they took supernatants from these cultures and then tested the supernatants using the IL-2 assay. So that was their experimental system. It looked just to show that the cells would react to this particular peptide. Then what they did was they took 100 billion B cell hybridoma cells and broke them open and solubilized them and then purified the MHC encoded molecule. And then they did binding assays. So we've done a little bit of this with the IL-2 binding, TCGF binding assay. So here we have increasing concentrations of the derivatized peptide. And here we have the number of molecules of the derivatized peptide that were bound in the equilibrium dialysis experiment to the MHC encoded molecule. And you can see that you get a saturation binding curve as you increase as you increase the concentration of the free. This is free, this is bound peptide. And now there's a method that's called scatter plot analysis, and that's shown in B here. There's only these are sort of simple experiments, and you only get 2 data points. You get at different concentrations, you get bound and you have free. So if you take the ratio of bound over free and plot it on the y-axis here, and then plot the bound molecules here, you'll get if it's a single affinity binding site, you'll get a straight line that comes a straight line that crosses the x-axis at the number of binding sites per cell. And so this is comes down to 0.8 micromolar. So basically it's a 1-to-1 ratio, and the slope of this line allows you to calculate the affinity constant, or the equilibrium dissociation or association constant. And in this instance, that it gave a KD of 2 micromolar, 2 times 10 to the minus 6 molar, which is a pretty high concentration that you have to get of the ligand to get a 50% saturation. So that's about here on the bound over free scale here. Just for references, most antibodies, their KDs are somewhere between 10 to the minus 6 molar and 10 to the minus 9th molar. And by comparison, the KD for IL-2 binding to its receptor is even higher affinity, lower KD of 10 to the minus 11th molar. So you don't need much ligand when you have a very high affinity constant in order to saturate the binding sites. So you have to imagine that inside the cells where this binding is really taking place, or it can also bind particularly onto the helper T cells onto the surface on the outside, you have to have a pretty high concentration of the peptide, the antigenic peptide. And so that's basically the data. And in figure 3 here, we have competition curves showing specificity, so that without any competitor, there is this amount of binding, and then it's broken down by the peptide itself. So I'm going to stop screen sharing there, and we go into the discussion of this paper. So that meant that the binding was specific. So it was saturable, it was specific. And that's what you want to find in these kinds of instances in order to be confident that what you're really doing, what you're looking at is a real phenomenon. And now in the discussion, they point out that, and they reference several other papers where people had tried to bind peptides to MHC molecules on cell surfaces, and they failed. And I mentioned that, and they mentioned that in the intro. And the reason they failed was because as they increased the number of cells in their binding assays, they got into a very high specific background. So you couldn't, the amount of specific binding and the amount of non-specific binding to sites all over the cell surface was just too much, and you couldn't really figure out exactly what was going on. So by using purified MHC molecules, the non-specific binding was much lower, and in their experiments they calculated out that it was like around 5% of the total binding was non-specific binding. The rest of it was specific, and so that was good. The other thing they had to do in order to see the binding, because it really depended on the sensitivity of the fluorescence assay as to whether they could detect it or not, they had to use a high concentration of the purified MHC molecules. And they used 10 micromolar. And they also had to use a high concentration of the derivatized peptide, and they used 35 micromolar to start with, and then they did different concentrations and so forth. They also discussed the fact that it was already known at that particular point in time that a given MHC molecule could bind more than one peptide. So it wasn't a one-to-one correlation. And they didn't really, nobody really knew what that was, why that happened, but that was a characteristic. And so it made the antigen presenting cell a very sort of like a garbage can. They could pick up all kinds of things and show it to the T cell. So subsequently, what happened in this field was in 1987, only 2 years later, there was two landmark papers that came out in Nature. The first author was Pamela Bjorkman, and then Jack Strominger and Don Wiley at Harvard. Pamela was a graduate student, and this was the first structure of an MHC molecule that had been reported. Happened to be class 1, as I recall. But that was just unbelievable because, as they say, a picture is worth 1,000 words. And so what the MHC molecule, the structure of it is that it's like there it is. It's like a hot dog bun. This is one part of the hot dog bun, and this is the other part of the hot dog bun, with the peptide in between, like the wiener. Mm-hmm. So once you could see that, and it shows to Goya how much human beings as scientists need to really understand things. They have to be able to look at it. From that point on, from 1987 on, then structures of the T cell receptor binding onto the MHC peptide complex came out, and things started to be much clearer in terms of all kinds of experiments have been done over the prior 10 years, 10, 15 years, trying to figure out what this so-called MHC restriction was all about, and what was the role of the MHC encoded molecules, what was this antigen processing presentation thing. So that basically broke it wide open. And from the standpoint of molecular immunity, we now have had firm data to depend on. So that's it for this one. And don't forget to like the video and subscribe to the channel, and click on the bell and go down and look at it and read this paper, essentially 1985. And so that's it. Thanks for coming, and come back next week.